طيب احنا يعني our last lecture for today اللي هي الانتيفيرالز يعني somehow خلينا نحكي a nice simple subject. Let me just alright. طيب انا بالعاده let's say انا يعني كل ما بدي اشرح عن الانتيفيرالز انا بحط هالسلايد which shows a number of deaths from each let's say viral outbreak يصير عندنا في العالم يعني ودائما هيك بعلق على in 1918 صار في a huge أو let's say a great outbreak for H1N1 أو اللي هي معروفة بالسبانيش فلو. تقريبا more than 50 million people died of that Spanish flu. And it's a continuous يعني ودائما كنت أحكي إنه يعني الاوتبريكس أو الفيرال اوتبريكس بتصير بس عمري ما توقعت إنها يعني ممكن تصير let's say in our lifetime وقت الكورونا. ف يعني this is something مش بس يعني بسنة 1918 بلشت هاي الاوتبريكس، حتى كانت كمان يعني يعني before خلينا نحكي the first millennia كان في huge outbreaks وكانت تقضي على أعداد كبيرة من الناس. طيب هلا before we start talking about antiviral drugs and those agents خلينا بس هيك نشوف this slide which shows you that as you all know and viruses are not living organisms. These are actually let's say for them to thrive and survive they need a host. All right, now this diagram shows you that the necessity for a host because they cannot replicate their genetic material. They cannot, let's say, they do not have the machinery to synthesize the proteins which gives rise to their coats and whatever organelles in them. So that is why they need a host. And when they integrate themselves in a host, they eventually integrate their DNA in the host DNA makeup or map. And they use the machinery such as for instance the ribosomes اللي بالhost, most of the enzymes اللي موجودة بالhost including the enzymes that they have themselves to eventually replicate and assemble themselves and after release they will move on to another host cell and so on. Okay. طيب يعني لو ترجعوا للليترشر the number of antivirals that are under investigation FDA approved are huge. لكن even though يعني مع كل هالريسيرش اللي بصير عليهم many of the data regarding the SAR for the antivirals خلينا نحكي a huge number of analogs اللي تصنعت والسار اللي مبنية عليهم mostly are either patented أو أحيانا بتكون let's say معلومات الشركات الأدوية ما بتفضي عنها لأنه ال continuous outbreaks of let's say viral infections بالنسبة لهاي الشركات هاي مصدر كبير أو مهم جدا للأموال. فعشان هيك if whatever information they have regarding the SAR potent inhibitors مش كله بيعلنوا عنه، مش كله they will publish it and you find it in literature. That is why what we know regarding the antivirals is not much. فعشان هيك احنا ما رح نحكي او الصار regarding the antivirals it's not let's say very well established for most of them. رح نحكي عن just let's say two subclasses that they inhibit اللي هم DNA polymerase و reverse transcriptase. وحتى كمان both of those targets the number of antivirals اللي موجودة still we cannot let's say map great SAR activity or structure activity relationship information from it. Okay, the number of analogs اللي رح نحكي عنها somehow limited structure wise في some variation يعني ما فيش مجال إنه مثلا نحكي إنه what is let's say very important for functionality how they let's say bind well يعني many of those informations اللي كنا نشوفها في مثلا البيتا لاكتامز والانتي بكتيريالز للاسف انها مش كثير well established in literature regarding the antivirals. طيب بس بدي اتاكد انه انا عم بعمل recording. Yes, good. طيب هلا رح نبلش من not from the first antivirals that were developed or discovered رح نحكي عن the one where it was considered a breakthrough, a compound or an antiviral agent that had great activity and gave rise to, let's say, many analogs later on. The analogs before رح نحكي عنهم في الآخر. مشكلة هاي الانالوجز في البداية اللي طوروها ما كانت لها great activity وكانت لها a lot of side effects and toxicities. Okay, only when it came to acyclovir, this is the one that gave rise to very good activity, very good selectivity, and for sure accompanying that much less side effects. هلا الاcyclovir لو انا مثلا بدي اصنفه structure wise شو بعتبره؟ Its structure is based on what?
هو نعتبره جوانسين انالوج.
Yeah, so it's considered a nucleoside analog. Okay, in particular, it is, let's say, synthesized or developed based على deoxyguanosine triphosphate. Okay, that's actually the bioactive form. يعني هو acyclovir is the one that does not have those phosphates. Those phosphates are added to the molecule after, let's say, it enters the virally infected cells. That is why acyclovir احنا بنعتبره a prodrug. طيب شو بيختلف structure wise عن الديوكسيجوانسين or guanidine? Yeah, so it has an acyclic structure. That is why it's acyclic or acyclovir, because it lacks the cyclic structure of this deoxyribose, and for sure it lacks this hydroxyl. We will show later how when missing that hydroxyl, it leads to eventually to what is known as chain termination and inhibition of DNA replication. The target for this drug is for sure a viral DNA polymerase. طيب, as I've mentioned, this compound is considered a prodrug, which means that it needs to be triphosphorylated to eventually give rise to the acyclovir triphosphate, which is the active form, or in other words, the one that binds eventually to DNA polymerase. طيب, what they found out, which is one of the main reasons why acyclovir يعتبر selective وبالتالي side effects قليلة أو much less مقارنة ب خلينا نحكي first antivirals that were developed. The first step, which is considered the rate limiting step, the first phosphorylation that occurs requires thymidine kinase, in particular the viral thymidine kinase. This step is not or cannot be, let's say, fulfilled by our thymidine kinases present in our cells. But that is why, or this is one of the reasons why acyclovir, after entering the cells, it will selectively be phosphorylated in virally infected cells. حتى لو دخل, let's say, our cells that do not have, let's say, viral infections or viruses inside them, cellular thymidine kinase, our thymidine kinase cannot, let's say, proceed with the first step, which means إنه ما راح يصير في عندنا triphosphate form, which is able to bind to the DNA polymerase. لكن the later steps can, let's say, occur via cellular kinases. Again, it only gets activated or selectively gets bioactivated in virally infected cells. Okay? When it enters the cell, when it enters the virally infected cells, after the bioactivation by the viral thymidine kinase to give rise to the monophosphate, and later by cellular thymidine kinases to eventually give the triphosphate, that acyclovir triphosphate can be incorporated into the DNA that is being built by DNA polymerase. Okay? To begin with, راح يرتبط عندي هالدرug بالDNA polymerase. Okay? So when it binds to DNA polymerase, معناها it competes with the already, let's say, normal nucleosides. Another thing is that when it binds to DNA polymerase that is already building the DNA, okay, do you think it can be incorporated into that DNA that is being built? انه هل مثلا هل ممكن انه لما لما يرتبط بالدي ان اي polymerase الدي ان اي، هل انه ممكن الدي ان اي polymerase يربطوا على the strand that is being built؟ هلا you are true but this leads to انه ما في شيء بيرتبط فيه، بس هو already ممكن يرتبط بالدي ان اي. يعني Look at this figure. This shows you the template DNA strand that the DNA polymerase is using to build, let's say, the daughter DNA. Okay? لاحظوا هون إنه when that acyclovir binds in, the process via which كيف بترتبط عندي النيوكليوزايد مع بعض, which is catalyzed by DNA polymerase, إنه الهيدروكسيل اللي موجود عندي على position 3 will attack الphosphate. Okay? And this entire pyrophosphate will leave, and this is how the bond, let's say, will be formed ما بين الهيدروكسيل of the nucleoside اللي already موجود عندي في الدوتر DNA, خلينا نحكي strand, والacyclovir. Okay? لكن exactly. لكن if another nucleoside is to bind in, because this one lacks the hydroxyl in here, حتى whatever a nucleoside will comes in, it will not be able, or the enzyme will not be able to bind it to it. But this is how it terminates the synthesis of the DNA. Okay, so it is considered a chain terminator because it lacks the hydroxyl, the nucleophilic hydroxyl at position 3'. Okay. طيب. Again, we've mentioned that one of the selectivity reasons, one of the reasons why acyclovir is considered a selective drug to virally infected cells, اللي هي إنه they have or they will be selectively converted into the monophosphate via the viral thymidine kinase. Only the viral thymidine kinase ممكن يرتبط فيهم ويحولهم للمونو. Later on, they will be converted into the tri, which is the ready form to bind to DNA polymerase. In cells that are not virally infected, okay, that do not have a viral thymidine kinase, حتى لو دخلوا هاي السيلز ما راح يصير لهم any kind of activation وما راح يرتبطوا بالDNA polymerase, our mammalian DNA polymerase. The other reason is that they get selectively uptaken by virally infected cells. The exact mechanism how it happens is not well known, but for some reason the viral infected cells may have, let's say, alteration in their permeability, alteration بالreceptors اللي بتدخل عنا الnucleosides. This is why they will get, let's say, selectively uptaken by those virally infected cells. And the third reason is that the selectivity of the acyclovir triphosphate to DNA polymerase, to the viral DNA polymerase, is way much more than our mammalian DNA polymerase. فحتى لو صار له bioactivation, all right, راح يفضل يرتبط بالviral DNA polymerase but not the human DNA polymerase. For all of those reasons combined is why acyclovir is a safe selective antiviral agent. اوكي طيب المشكله الرئيسيه اللي كانت بالاcyclovir اللي هي الاورال bioavailability. If absorbed, only up to 30% of that dose will get absorbed. They tried to modify the structure in order to enhance the oral bioavailability. And this is what resulted in valcyclovir, one of the analogs that proved to be successful in enhancing the oral bioavailability, or an analog that had a much better oral bioavailability is valcyclovir. Look at the two structures واحكوا لي شو الفاريشنز ما بينهم.
ضفنا شو اسمه حمض اميني اللي هو
Yep. This which resembles الفالين.
الفالين.
Yes. هلا acyclovir in terms of absorption much better than acyclovir. Do you have an explanation for that? Exactly. So it can be selectively uptaken, or it binds, let's say, or it has good affinity to di and tri, let's say, peptide transporters. But being actively transported is what has increased its absorption or its bioavailability relative to acyclovir. Okay? هلا is valacyclovir ready for phosphorylation? Yep, لازم يصير له hydrolysis here, all right, to expose that hydroxyl. Then it can bind to the viral thymidine kinase to get phosphorylated. Okay? الجانcyclovir شو بيختلف عن الاcyclovir؟ Yeah, we have a hydroxymethylene group that is added in here. All right. هلا the explanation for how ganciclovir relative to acyclovir has better, let's say, better oral bioavailability is because it is, or it has more, let's say, water solubility. فالabsorption بيكون أفضل. And add to that, طبعا هاي in, let's say, in some references, إنه when you have, let's say, those two hydroxyls together, معناها لما الثايميدين كاينيز يرتبط فيهم بيكون عنده في two groups ممكن يربط عليهم الفوسفات rather than just one relative to acyclovir. يعني in other words الريت والextent of المونوphosphorylation for ganciclovir أحسن. تمام؟ طيب another analog انه هاي ممكن يصير لها let's say you mean it may bind to whatever nucleotide that will be added later. لا هي عم تحكي عن الchain termination انه هل can I consider الجانسايكلوفير a chain terminator زي الايسايكلوفير؟ لا هلا ممكن انه هاي يصير لها او it might get let's say after being phosphorylated attacked by the hydroxyl with the nucleotide that the acyclovir or ganciclovir will be added to. هلا you are right in terms of ممكن انه هاي الهيدروكسيل might let's say act as a nucleophile where it will attack the phosphate in any incoming nucleotides. بس لانها rotatable bond, فالchances for it to be in the right position, right distance from whatever phosphate of any upcoming أو incoming nucleotides أو nucleosides ما راح تكون الchances أقل بكثير إنها تعمل attack مقارنة لو كان أنا عندي a rigid structure with a hydroxyl that is rigid, fit exactly in the right position, the right distance from the phosphate اللي يفترض إنها تتفاعل معها. طيب الفامسايكلوفير, all right, which is also even a prodrug of pencyclovir. Both of those analogs, where one is a prodrug of another, they are also analogs of الacyclovir. طيب شو الchanges اللي انعملت فيهم؟ Look at the structure of acyclovir والpencyclovir. Yeah, so relative to ganciclovir, it resembles more ganciclovir. And we have, let's say, a hydroxyl or a carbon substituted oxygen. Okay, and as for famciclovir, it requires bioactivation via esterases حتى نكسر هاي الester bonds later on by aldehyde oxidase in the liver to convert this ring system into, let's say, this one. All right, all of those. For sure, they need triphosphorylation. Otherwise, ما راح يرتبطوا في DNA polymerase. هلا after the introduction of acyclovir and all of those analogs into the market, they found out some strains of viruses have developed resistance. يعني in particular, for instance, some strains of herpes simplex, they found out after, let's say, a while, they developed resistance against those drugs. And that resistance, they stopped producing a thymidine kinase, which is, as we have mentioned, very crucial for a conversion of acyclovir and pencyclovir into the di, later on into the mono, which will later on be triphosphorylated حتى يرتبط في DNA polymerase. That step, that first monophosphorylation step, does not happen via the cellular thymidine kinase or kinases. فإذا ما صارت عندي أول step ما راح يصير في عندي triphosphorylation، ما راح يصير bioactivation لهاي الدrugs بهاي resistant strains of viruses، وبالتالي ما راح يصير في عندي any binding to DNA polymerase. طيب برأيكم كيف حاولوا، طبعا بالنهاية حلوها، يعني how you think their approach was حتى إنهم يحلوا هاي المشكلة structurally. You are true, you are right لأنه simply شو رح يصير عندي؟ يب بصير لها hydrolysis in vivo. لكن أنا ممكن أعطي إشي bioisosteres إشي resembles it لكن does not undergo hydrolysis. So this is what they've done. Okay, they've developed, let's say, an analog of deoxycytidine monophosphate, which is Sedovir, okay, where they've added not a phosphate group وإنما a phosphonomethylene group. Okay, يعني حطوا بين الأكسجين وبين الفوسفات أو بين الفوسفورس أميثايلين. طيب طبعا later on this group can be easily di and triphosphorylated. So how come this one does not undergo hydrolysis and this one undergoes hydrolysis? هلا if let's say water will attack الفوسفات ممكن تنكسر عندي هاي البوند, right? But here I have a carbon. هاي البوند ما بين الفوسفات والفوسفورس والكربون is not let's say a weak bond, does not break. فحتى لو صار في عندي attack على هالفوسفات ممكن الهيدروكسيلز تطلع. alright, لكن ما راح يصير في عندي let's say breakage ما بين هاي الفوسفات أو الفوسفورس والكربون. تمام؟ Okay. So Sedovir relative to acyclovir, because it resembles a monophosphate form, it will directly be diphosphorylated and triphosphorylated, giving rise to the bioactive form. طيب نحكي هلا عن other analogs which are considered also inhibitors of DNA polymerases, لكن different than the one we've seen before. Idoxuridine, trifluorouridine, vidarabine. These are actually the first antivirals that were developed. They were actually developed to treat the cancer, لكن they found out that they actually have antiviral activity. Those were the first antivirals to be used. لكن لأنهم ما كان لهم selectivity عالية, alright, so that is why they had much more or a lot of toxic and side effects, which were later replaced by acyclovir. And these are actually the first antivirals that were approved. All right. هلا طبعا for sure these are nucleoside-based antivirals. The variations, if you can, let's say, see in here, for instance, في حالة idoxuridine, they replaced the hydrogen with an iodine or a trifluoromethyl. Okay. هلا how they inhibited DNA polymerase, how they inhibited DNA synthesis, because when they integrate or when they bind to the DNA polymerase ما راح يصير في عندي efficient base pairing. That is why this will not, or this will lead to inhibition of DNA synthesis. آخر antiviral that is considered an inhibitor of DNA polymerase اللي رح نحكي عنه اللي هو الفوسكارنت, which has this structure. It's an analog of what? Structurally, it's an analog of what you think. بيشبه مين structurally?
بيشبه الفوسفات جروب بس عليه كربونات وصوديوم.
تمام. So it is a phosphate group analog where one of the hydroxyls is replaced with a carbonate. Okay, الفوسكارنت الابزوربشن له سيء, التوكسيسيتي له relatively عالية. Usually this drug is kept in strains that are considered resistant to the already available antivirals. Yeah, it binds to DNA polymerase also and inhibits the DNA polymerase. ننتقل هلا ل another group which are the reverse transcriptase inhibitors. هلا you all know طبعا هاي شيء موجود في الانترودكشن, يعني for sure read it. I'm sure انه غطيتوها بشكل كبير بالمادة الميكروبيولوجي, بس احنا يعني this is not our scope of this course, ف يعني مش راح احكي عنها, but you already know that viruses could have, or their genetic material could be DNA or RNA. Those viruses that have, let's say, their genetic material as RNA, for them to use the host and replicate, they need to let's say synthesize DNA out of its RNA genetic material. And for that process, they require an enzyme that is called reverse transcriptase, which changes or which synthesizes DNA based on its RNA material. But that is why when they discovered that the reverse transcriptase enzyme was the target for many research efforts, لأنه our, let's say, cells do not have that enzyme. بالتالي, if you would inhibit this enzyme, you would inhibit, let's say, any viral infections from those, let's say, viruses without affecting or without having any side effects associated with them, لأنه we do not have reverse transcriptase. Okay, a very famous, let's say, virus that is considered or that has this enzyme اللي هو الAIDS. Most of the AIDS treatments are based on inhibition of reverse transcriptase. Let's see if this, or if I can show you. Good. So this is a video that shows you reverse transcriptase bound to DNA, where it will later zoom in على where, let's say, one of those analogs can bind in. اللي راح تفضلي. Yep, definitely. هلا for those, let's say, reverse transcriptase inhibitors, راح نحكي عن those drugs which are considered nucleoside based and also non-nucleoside based inhibitors. It should zoom in. I'll download, let's say, or upload this video for you. As it shows very nice, let's say, information about the binding site. طيب راح نحكي هلا عن أول group, which are the reverse transcriptase inhibitors that are based on nucleosides. And the first one is zidovudine. Okay, look at the structure and tell me it resembles what? Yeah, they replaced it with what? Yep, exactly. It's the azide. Yeah. So it's based on deoxythymidine, where the hydroxyl is replaced with N3 or an azide group. Here, this represents the structural the structure of the N3 group. Is it considered a good nucleophile? Mm-hmm. Yep. Exactly. Yeah. So in this resonance form drawn in here, that negative charge, as you can see, is right next to an atom that is positively charged. في عندي huge electron withdrawal via, let's say, via inductively and even via resonance. يعني I can, let's say, draw another resonance form for this compound, where that negative charge is delocalized between, let's say, two of those nitrogens. All right, so that is why it's a very bad nucleophile, very similar to the ones we've seen for the DNA polymerase inhibitors. They do bind to reverse transcriptase, but they eventually will lead to termination of the DNA synthesis. Okay, here are, let's say, other examples: lamivudine, emtricitabine, which are based on deoxycytidine, and as you can see in here, they removed the hydroxyl or any, let's say, other substituent attached to carbon 3, and they've substituted carbon 3 with a sulfur. صح, الsulfur تعتبر a good nucleophile, لكن still it is far away from the phosphate of the incoming nucleotide. Very similar to the ones we've already discussed, it is also considered a chain terminator. Okay. هلا, do you think those are prodrugs? Or they are active as drawn in here? Exactly. for them to bind to reverse transcriptase واللي بيشتغل بنفس الطريقة as DNA polymerase, it requires the triphosphorylated form. هلا different from acyclovir, the entire phosphorylation, mono with di triphosphorylation of those drugs happen, let's say, via or can let's be catalyzed all of the steps, not just the di and tri, also the monophosphorylation via the cellular thymidine kinases. Okay. Second, you can consider the main reason for their selectivity because they have selective binding affinity to an enzyme that we do not have, which is reverse transcriptase. The adenosine is also an analog that is considered a nucleoside reverse transcriptase inhibitor. It's definitely a prodrug that not only it requires a triphosphorylation, but it needs to be converted, or this inosine ring needs to be converted into the corresponding adenine. Right. Here are, let's say, other drugs. Oh, before we move on to this, do you think the adenosine is considered a chain terminator? Yeah, because that sugar part, that the sugar part in it, it lacks the hydroxyl at position 3 prime. These are, let's say, other analogs. أنا فقط بدي أعلق عليهم الأديفوفير dibivoxyl with tenofovir disoproxyl, which are considered a pro-drugs. Very similar to cidovir. It's already monophosphorylated, as you can see in here. These are ester forms. اللي بيصير لهم ببساطة إنه when that ester is hydrolyzed, eventually it will give rise to the phosphonomethylene analog, which will then be di and triphosphorylated. The esterification here in both of those analogs is for the purpose of increasing the water solubility and absorption. Sorry, مش water solubility, lipophilicity أسفة and absorption. Okay. آخر جزئية رح نحكي عنهم اللي هم non-nucleoside reverse transcriptase inhibitors. كل شيء مذكور هون بالسلايدز مش ضروري منكم تعرفوه. يعني you will not be asked about their structures because as you can see they are completely variant. الإشي الوحيد اللي بيجمع بينهم كلهم إنهم hydrophobic compounds. Do you think إنهم competitive inhibitors ولا allosteric inhibitors of reverse transcriptase? Allosteric, exactly, because if you look at their structures, they do not resemble the nucleosides. They are actually considered allosteric binders. طب مين من عندهم أو مين رح يكون في tendency أكثر إنه يصير في emergence of resistance against them? Those non-nucleoside reverse transcriptase inhibitors ولا الnucleoside? Yes, because for the nucleoside inhibitors, they bind into a site where many of the amino acids اللي بتكون موجودة عندي are actually conserved, important for the catalysis that the enzyme carries. Right? لكن for those ones, because they bind to allosteric positions, as you can see in here, that is why ممكن يصير في عندي أو ممكن يصير في simple mutations without affecting the function of the enzyme. The image you see in here represents the reverse transcriptase. This is, let's say, the DNA RNA strands. The site B represents, as you can see in here, a prosthetic. Sorry, it resembles the position where nucleosides usually bind in, and where, for instance, the AZT binds in. And as for C, this is the allosteric site where one of those non-nucleoside reverse transcriptase inhibitors bind in. Very close sites to each other. Lakin one is considered, let's say, the active site of that enzyme, and another is allosteric. Okay, so as I have mentioned, the structures of those drugs, the interactions that they form, is definitely not something to memorize. The variations in structure between them تماما أو مختلفين تماما وتماما في عندي variations كبيرة. The only thing, as I have mentioned, that combines them or that distinguishes them is that they are hydrophobic in nature. Yep. هلا for instance What do you mean ال O is clear? But you have, let's say, a third butyl group. You have a methylene in here. Yes, a third butyl right next to a methylene also. Generally speaking, those, let's say, esters are actually more lipophilic relative to the parent drug. All right. Any questions so far about the antivirals? Which ones? The AZT, you mean? Yes, they are given orally. Yep. الأزيدو group, for instance, is a lipophilic group. It is considered, even though it is, let's say, charged, لكن the overall charge صح negative, لكن it's a polar functionality, لكن generally speaking that group increases the lipophilicity. But in terms of absorption, they are actually well absorbed when taken orally. Any question, Karam?